Hello students, in today's class we are going to learn about endoplasmic reticulum. So endoplasmic reticulum along with other organelles such as Golgi complex, lysosomes and secretory vesicles form a system which is known as endomembrane system. So the endomembrane system of eukaryotes consists of the endoplasmic reticulum and the Golgi complex that are involved in protein synthesis, processing and sorting, the endosomes that carry and sort material entering the cell, the lysosomes that are involved in digestion of both ingested material and unneeded intracellular components, and the transport vesicles that are involved in transportation of membrane lipids and membrane bound proteins to their proper destination in the cell, and they also carry soluble materials destined for secretion. Thus, the endoplasmic reticulum along with Golgi complex, endosomes, lysosomes and the vesicles are connected with each other, forming a dynamic membrane system which is known as the endomembrane system. Coming to the structure of endoplasmic reticulum, endoplasmic reticulum is organized into a network of branch tubules and flattened sac-like structures that cover the entire cytoplasm. The tubules and the sacs are interconnected and the membranes are continuous with the outer nuclear membrane. Thus, the endoplasmic reticulum and the nuclear membrane form a continuous sheet enclosing a single internal space, which is known as endoplasmic reticulum lumen or the endoplasmic reticulum cisternal space. The rough endoplasmic reticulum consists of large flattened sheets. These are also known as transitional elements, which are involved in synthesis of transition vesicles that shuttle lipids and proteins from the endoplasmic reticulum to the Golgi complex. The smooth endoplasmic reticulum has tubular structure forming a lace-like network near the cellular periphery. The same points are mentioned in this slide for your reference. Coming to the function of rough endoplasmic reticulum, they are basically involved in three main functions. Firstly, they are involved in biosynthesis and processing of proteins. So ribosomes, they are basically normally present in the cytosol. However, during protein synthesis, they will attach to the cytosolic phase of the endoplasmic reticulum, and the newly synthesized proteins will enter the endoplasmic reticulum membrane during the translation process. Endoplasmic reticulum is also involved in post-translational modifications. These modifications include glycosylation, which is important for sorting of the protein to their proper destination. And the pattern of the end-link glycosylation is used to indicate the extent of protein folding so that the protein leave the endoplasmic reticulum only when they are properly folded. Disulfide bond formation also takes place within endoplasmic reticulum, which is another important parameter for proper protein folding. Thirdly, quality control is the third function of the endoplasmic reticulum. So those proteins which are improperly folded are basically targeted for degradation by cytosolic proteasomes before they can move to the next organelle, that is Golgi complex, for further processing. So the proteins that are synthesized on the rough endoplasmic reticulum, they have three destinations. Either they are proteins that are secreted outside the cell, or they are components of the integral membrane, or they are soluble proteins of the cellular organelles. Next, coming to how the ribosomes attach to the surface of the rough endoplasmic reticulum, and how does the newly synthesized proteins or polypeptides will enter into the lumen of the rough endoplasmic reticulum. We will try to look at the mechanism that allows this transportation. So there is a hypothesis which is known as signal hypothesis that would explain completely the mechanism that the ribosomes follow in order to attach to the outer surface of the rough endoplasmic reticulum, and then how the proteins are transported into the lumen of endoplasmic reticulum. Now discuss the mechanism of signal hypothesis. So here we can see the messenger RNA molecule, which has the 5' cap and a poly A modification. So the ribosomes will scan the messenger RNA from the 5' end and will begin the translation process as it reaches the start codon, AUG codon. So a short stretch of peptide is synthesized containing the signal sequence. As soon as the signal sequence is synthesized, it is bound by the signal recognition particle, which then also binds to the ribosome. The signal recognition particle is then bound by GTP, and it inhibits the translation process and targets the ribosome to the rough endoplasmic reticulum by binding to the SRP receptor present on the endoplasmic reticulum. After this, GTP is hydrolyzed and SRP is released, and the ribosome binds to the protein translocation complex on the surface of endoplasmic reticulum. Once the SRP is released, protein synthesis is resumed, and as the protein is synthesized, it will enter through the protein translocation complex into the lumen of endoplasmic reticulum, completing the protein synthesis process. So as the signal sequence enters into the lumen of endoplasmic reticulum, it is recognized by a specific signal peptidase that chops off the signal sequence, and then the mature polypeptide chain will undergo folding to form the final structure. The same points are mentioned on this slide for your reference. Next, we will discuss about the glycosylation reactions that take place in the lumen of endoplasmic reticulum. So as such, there are two types of glycosylation reaction. One is the N-link glycosylation reaction, which involves the nitrogen of the amino acid asparagine residue. While in O-link glycosylation, there is involvement of the oxygen of the hydroxyl group of serine or threonine amino acids. So the O-link glycosylation reaction takes place in Golgi complex, while N-link glycosylation reaction takes place in the endoplasmic reticulum. We now discuss about the different steps of N-link glycosylation reaction that takes place in the rough endoplasmic reticulum. So the dolicol phosphate molecules over here act as a carrier molecule for synthesis of a core polysaccharide on its surface. The process starts with addition of two residues of N-acetyl glucosamine, followed by addition of 5 residues of mannose sugar, resulting in total addition of 7 sugars on the surface of dolicol phosphate. Then we have a translocation step wherein the dolicol phosphate molecule flips so that the core that is synthesized so far is now present towards the luminal face of endoplasmic reticulum, and the rest of the modifications or synthesis of the core oligosaccharide will take place on the luminal face of endoplasmic reticulum. So here we will have addition of additional mannose and glucose residues by making use of dolicol phosphate derivatives as donors. Now, this leads to construction of eventually 14 sugar core polysaccharide on the surface of dolichol phosphate. Once the 14 sugar core is synthesized, next we have transfer of this core onto the surface of the nitrogen of the asparagine amino acid of the polypeptide that is entered or is synthesized by the ribosomes such that it enters into the lumen of endoplasmic reticulum. Once the 14 sugar core is transferred onto the asparagine residue of the polypeptide chain, we have cleavage of additional sugar residues, and only 5 sugars are retained on the core polysaccharide, which consists of 2 residues of N-acetylglucosamine and 3 residues of mannose sugar. Further modification will take place, which differs from protein to protein, which includes addition of different types of sugars and different numbers of sugars depending on the protein. At the end of the process, we will have generation of the dolichol phosphate molecule again. The released dolichol pyrophosphate molecule will then be translocated so that it is present on the cytosolic phase of endoplasmic reticulum, and then the phosphate group is hydrolyzed or removed, and that leads to regeneration of dolichol phosphate molecule, which can be recycled, and the entire process can continue. The same points are written in this next slide for your reference. So the next slide discusses the modification or processing of newly synthesized proteins and how it involves the role of chaperone proteins. So here we have a chaperone protein that is cannexin, which provides an environment for proper folding of the protein. So those proteins that are correctly folded are further transported through vesicles into the next organelle, that is Golgi complex. However, if the proteins are not folded properly, then they are recognized within the endoplasmic reticulum, and improperly folded proteins, instead of being transported to the next organelle, they are basically transported to proteosomes where they are degraded so that the amino acids can be regenerated and recycled. So in this way, the endoplasmic reticulum plays a role in quality control, ensuring that only properly folded proteins are allowed to leave the endoplasmic reticulum. Next type of modification or processing that takes place in the endoplasmic reticulum is formation of disulfide bonds within the polypeptides. So these disulfide bonds can be intra-chain, that is formed within a single polypeptide chain, or they can be also interchain, wherein they are involved in formation of dimers, tetramer-like structures. So the amino acid that is involved in formation of the disulfide bonds is amino acid cysteine, which contains the SH group, also known as the sulfhydryl group. So the oxidation reaction between two such R groups of the cysteine amino acid will lead to loss of hydrogen and formation of internal sulfur-sulfur bond, which is known as the disulfide bond. This plays an important role in interchain as well as intra-chain protein folding. So coming to, in summary, the function of rough endoplasmic reticulum involves synthesis of proteins on the ribosomes of rough endoplasmic reticulum, and modification and processing of the newly synthesized proteins by glycosylation, folding of the protein, by formation of the disulfide bonds, and also ensuring the quality control process. Lastly, it is also involved in transport of the protein. Next, we will discuss about some of the functions of smooth endoplasmic reticulum. Firstly, it plays an important role in drug detoxification. So it involves an enzyme catalyzed hydroxylation reaction because the addition of hydroxyl group to the hydrophobic drugs make them more soluble and easier to excrete from the body. So example is involvement of cytochrome P450. So RH here represents a drug that needs to be detoxified. NADPH molecules are involved and oxygen will be used as a donor to add a hydroxyl group to the drug. Now, addition of hydroxyl group to the drug will make it more soluble and it can be easily removed from the body. Another example includes the use of enzyme aryl hydrocarbon hydroxylase, which is also synthesized in the smooth endoplasmic reticulum. This complex is involved in metabolizing polycyclic hydrocarbons. So these are basically pesticide molecules that are made up of two or more benzene rings that are linked with each other. These are often toxic, and here also the enzyme is involved in the hydroxylation reaction, which will again increase the solubility of the molecules in water, and that is required for removal of the molecules from the body. However, the oxidized products are often more toxic than the original compounds. Then coming to the next function, that is carbohydrate metabolism. So within the liver cells, we have storage of glycogen molecules, and the glycogen molecules are need to be converted into glucose molecule before they are distributed throughout the to other cells. So here the after breakdown, it gets converted into glycogen gets converted into glucose 6 phosphate molecules, and the enzyme glucose 6 phosphatase is required to remove the phosphate, generating glucose molecules that can be then transported into the other cells through the blood, where it can be used as an energy source. And then it is also involved in calcium storage. The sarcoplasmic reticulum that is found in the muscle cells is an example of smooth endoplasmic reticulum that specializes in storage of calcium ions. So these calcium ions are pumped into the endoplasmic reticulum by making use of ATP dependent calcium ATPase, so that they can be stored within the endoplasmic reticulum. And in response to some kind of an external stimulus or signal, the calcium ions are released, and this is recognized again as a signal and helps in muscle contraction. Smooth endoplasmic reticulum is also involved in biosynthesis of steroids. So it is a site for biosynthesis of cholesterol and steroid hormones such as cortisol, testosterone, and estrogen. So the enzyme involved is hydroxymethyl glutaryl coenzyme A reductase, which is required for cholesterol biosynthesis. And this enzyme is present in large amounts in the smooth endoplasmic reticulum of the liver cells. Here it is involved in synthesis of cholesterol and further its conversion into the steroid hormone by the hydroxylation reaction. So the endoplasmic reticulum, smooth endoplasmic reticulum, not only plays an important role in cholesterol biosynthesis, but it is also involved in synthesis of membrane phospholipids. One of the common example of phospholipids is phosphatidylcholine molecule. So coming to the structure of phosphatidylcholine molecule, it consists of a backbone structure that is made up of glycerol. To the two carbons of glycerol, we have attachment of fatty acid chains in an ester bond. And to the third carbon, we have attachment of a phosphate group. The phosphate group, the oxygen of the phosphate group is further involved in bond formation with a choline molecule in case of the lipid phospholipid phosphatidylcholine. So here the phosphate group along with the choline molecule or any other substituted group that might be attached to the phosphate group forms the polar head molecule, while the fatty acid chain along with the glycerol backbone will form the non-polar tail portion that is buried in the bilayer, while the head groups will be present either towards the cytosolic or the extracellular phase where they can interact with the water molecule. So each step of the synthesis of phospholipid molecules will take place on the ER membrane that have their active site facing the cytosol. And in the cytosol, we have all the requirements or metabolites that are required for synthesis of phospholipid. So phospholipid synthesis will occur exclusively in the cytosolic phase of the endoplasmic reticulum membrane. Since fatty acids are not soluble in water, they are transported to the ER by a fatty acid binding protein in the cytosol. And after arrival in the ER membrane and activation of coenzyme A, acyl transferases will successively add first two fatty acid chains to the glycerol phosphate molecule to produce phosphatidic acid. And then further we will have addition of the different types of substituents. Either you can have addition of a choline molecule in case of phosphatidylcholine. Other substituents will result in generation of different types of phospholipids such as phosphatidyl serine, phosphatidyl ethanolamine, and so on. So that's it about different functions of smooth endoplasmic reticulum. Kindly like, share, and subscribe if you like the content.